Yeah, thank you very much for this kind introduction. And I think all of you have realized that in like many other medical subdisciplines, also in anesthesiology and also in critical care medicine, molecular biology is becoming stronger and stronger. And even though at this stage it appears that most of the questions that we can answer by looking at physiology, I think there lies great promise also in investigating ventilation-induced lung injury and of course also sepsis-related phenomena and so forth on a molecular level. And what I try today is to give you an overview over some concepts in this area. And of course the first idea is to give you some background that will help you to understand what will be presented later on in this conference, but of course what also will help you to understand the papers that you will read. And also in journals like anesthesiology, there are more and more papers that really focus on molecular details. So I think it's very important to have a basic understanding of what's going on. Later on, of course, I think we will also realize that the knowledge that we develop on a molecular basis, also in anesthesiology, for instance, can be applied in terms of testing, of monitoring, and later on maybe in terms of therapies. And of course, this is basically very far-fetched, and I know that many of you are sort of disappointed by all the sepsis trials or anti-cytokine trials that failed. So I think there is some sort of disillusionment now that we say, well, it's not really worth pursuing these things. But I think this is wrong, because, for instance, if you look in an area like cancer, where people worked for many years really intensively on signaling mechanisms, and, for instance, tyrosine kinase inhibitors have been very long on the agenda in cancer. And only very recently, we have the first tyrosine kinase inhibitors that can be used in patients. So it's certainly a very long way, and we should not be disheartened if we are not successful in the first place. So, of course, the definition of molecular biology is a very broad one. Basically, it's all the biology we can do with molecules. So one definition reads, molecular biology is a study of biology at the level of molecules rather than tissues or cells. So what you will hear today is really a lot about molecules. I will never leave the cell, really, in what I will show you. Most of it will have to do with molecules. And also, I will have very little time, really, to focus on the consequences. I will talk a lot about signaling pathways and the molecules which are involved. And clearly, I think that they are important in ventilator-induced lung injury and sepsis. But I will certainly have no time to go into the details here. So this will be a very fundamental lecture that tries to give you some background. So at the end, I think you should know something about the constituents of the plasma membrane and some new developments that have led to the deeper understanding of lipid rafts and Caveolae. Then stay a little bit with the plasma membrane and show you that some of the constituents of the membrane really play a very important part in signaling. Then comes to one particular signaling mechanism that is signaling by extracellular proteases that sit on the cell. From there, we see how transcription factors become activated, and then later on see how this then leads to gene transcription, and look a little bit at the roles of histone in this case. So one of the basic questions, of course, that we have is, we have a cell and we have to stimulate that cell. And basically everything that happens to a cell is a sense from the cell as an input, and there are different ways for cells to sense that. It can be sensed, of course, by receptors, but it can also be sensed as a cytoskeleton if the cell is deformed and so forth. And then, of course, the cell has to produce an answer, and in many instances there is an output, so the cell is delivering another signal or giving a response. And I think if I try to come along and grasp all these really puzzling details in molecular biology, it's always good to start to get some boxes where you can put things in. And of course, this is a very easy kind of box scheme, where you just look at things starting at the plasma membrane. So this could be receptors, but as you will see, also lipid rafts and Caveolae. Then you have the signal transduction steps, and then you have the gene induction. So these are all steps that can be viewed separately. Also, in most of the schemes you will see, of course, they are very much interconnected. And if you look into signaling, I think we have two principally different ways. So one is indicated by this arrow. So you stimulate a cell, be it by a receptor, for instance, and then this cell produces an immediate answer with no gene transcription. So like, for instance, glycogenolysis being turned on if epinephrine is binding to liver cell. Or then we have another approach, a way which takes somewhat longer, because this involves gene transcription, and then genes are turned on, your gene transcription, and then those gene products may, for instance, be released from the cells. So I like to start now with the very first step at the plasma membrane, because also in this area there have been some very interesting developments recently. And I think this will all of you remind you on your textbook. So here we have two of the very basic constituents of the plasma membrane. You all know the plasma membrane is a bilayer, and these are two very important phospholipids. So we have phosphatidylcholine here, and we have sphingomyelin here, right? And you see this is a glycerol backbone, and here we have two fatty acids, and this is a phosphocholine moiety. And I think this molecule has been around for a long time, and many people are sort of familiar with it. Now there is a new, well, not a new class, but a class that recently has become more and more important in our thinking, our understanding of the cell, and those are the sphingolipids. And one important sphingolipid is sphingomyelin, as you see here. And you can really see, if you look at the structures, the structure is not really so much different of sphingomyelin from the phosphatidylcholine. You have also two fatty acids here, and this structure is not the glycerol backbound, but it's pretty similar to it, although this is an amide bond, really. And here again, there is a phosphocholine moiety. So these structures are pretty similar, and as we know, and I'll give you an example later on, we know that this is not only a molecule that makes a cell membrane, that builds a cell membrane, but this can also start signaling. The same is true for sphingomyelin. I come to that in a moment. Another important issue here is that the distribution of the sphingolipids in the cells is not homogeneous. It's not homogeneous in one way, and that has been known for some time. It's not homogeneous across a bilayer. So we know that most of the sphingomyelin inside in the cell membrane is in the outer leaflet of the membrane. And the same is true for phosphatidylcholine. And on the inside of the cell, we have rather more phosphatidylethanolamine and phosphatidylserine. So the serine and ethanolamine moiety is sitting here. And they have a negative charge, and so the inside of the cells is negatively charged because of this, partly because of those negative moieties here. So that's what we have known for some time, that we have a heterogeneity in sort of the vertical distribution of the phospholipids. What we have recently come to understand is that there appears to be also a difference in the horizontal arrangements of the phospholipids. So there appear to be islands of phospholipids of different kinds, and these islands have been termed lipid rafts. So this is a raft, and the idea is that these lipid rafts are floating in the other lipids. And these islands, they are very rich in sphingolipids like sphingomyelin. So here you see an atomic force microscopic picture of such lipid rafts, so however in an artificial membrane. And all those are sphingomyelin patches, so to say, on a phosphatidylcholine background. And the idea is that to start with, we may always have some rafts, and these rafts are characterized, of course, by the content in sphingolipids. And since sphingolipids have a higher melting point, they are stiffer. So these are areas in the membrane which are more rigid, and therefore it makes sense that these are preferable areas where the cytoskeleton connects to. And another concept that has emerged from that is that those lipid rafts, they can bring together signaling molecules. So we know, for instance, the T-cell receptor and also the toll-like receptor is really a complex of many, many molecules coming together. It's not only one molecule, it can be like 10, 15 molecules coming together and working on the same answer of the cell. And these molecules are thought to come together in those lipid rafts, are contained in those lipid rafts to provide an environment where they can come together and signal together. And a very special form of those lipid rafts are caveolae. And these caveolae have been known for many, many years. And this is a picture from a medical textbook of pulmonary medicine from Netta. Many of you will be familiar with it. And this is a book, this was made in 1984. So we are very well aware of the fact, or have been for a long time, that we have those caveolae. You can see here in some magnification, this is an endothelial cell lining in the lung, for instance. And you see that they are full of caveolae if you look at that. So these are those flask-like evaginations, as you can also see here. And they may or may not have such pelliculae on top of them, and the meaning of them is not very well understood. And for many years, really, we knew them, but we never really understood what they meant, what they were doing, really. And there are now two concepts emerging, what the role of those caveolae can be. And one of them is even indicated here, so I think this one, you can see there is one vesicle that obviously has been budding off from the caveolae. And one thing we know what these vesicles are doing is they transport albumin. So they can transport albumin from the vascular side to the interstitial side. And maybe, that's something we're just trying to think about, is that maybe some of the trials in intensive care medicine with albumin, and where you give additional albumin, can be complicated by such a mechanism, because that albumin could be transported across a barrier and then create a very high oncotic pressure, and by this mechanism enhance edema formation in the tissue. Another concept, or that has greatly improved our understanding, is the discovery of the molecule that very much defines those caveolae. So here you see a lipid raft, so this is rich in single lipids. And the concept now is that the binding of a molecule which was termed caveolin, because the caveolae were known, and when they purified the caveolae, they recognize this protein. So it's called caveolin. And there are three of them, three isoenzymes, caveolin 1, 2, 3. So if this protein binds, you can see in such a fashion, so intercalating here in between the two lipid bilayers, if it binds, then you get this sort of flask-like formation out of the lipid rafts. And of course now, the question is, apart from the transport of albumin to the cells, what can this mean? And just to give you one example, again, we think that this is a very important starting point for many pathways in signal transduction, for many pathways that lead to important end products in the cells. And one of them is NO, nitric oxide. And I'm sure that most of you will be aware of the important role that NO plays in all parts of biology. So of course it can relax airways and vessels, but it can also be pro-inflammatory if present higher concentrations and so forth. There's a great literature, of course, on NO. And one of this work has shown, and this is a work from, I think, 2003, that shows that NO can also contribute to ventilator-induced lung injury. And at least in that study, people said that this endothelial NO synthase was responsible in that study, even though recently they appeared another study that said another isoenzyme may be important. But I think that's not the point here. What is important now here is that this enos, the endothelial form of the NO synthase, is localized to the caveolae, and inside the caveolae, it's binding to caveolin 1, and that is shown here. Those are those yellow flags or whatever. So the caveolin 1 is binding the endothelial NO synthase, and as long as this binding occurs, the endothelial NO synthase is not active. That is indicated by those breaking lines here. And then, of course, later on, you can have stimulation, you can have phosphorylation of the enos or a binding of calcium to activate that. But in the present form, in this form, with caveolin being bound, it's inactive. However, it also shows that to get the enos to the membrane, you have to have caveolin, so it has to be there, it has to be able to bind there. So just to give you one example how this can also be of interest, and if you look at ventilator-induced lung injury, for instance, is that we know that if you ventilate the lungs, also some part of the vessels get stretched. Some of them get compressed, but the extra-alveolar vessels can get stretched. And here you see vessels in a rat lung, and these were pictures done from Wolfgang Kübler in Berlin in a study together with us. And this is a staining for NO. So this was a fluorescent dye that indicates the presence of NO in the living tissue. Okay, so this is a vessel, and you can see here the vessel has expanded, just like it would also do during ventilation. And you can see here these are spots of NO that are turned on by stretching those vessels. So we can clearly see that stretching the cells increases production of NO. And interestingly, these spots, particularly this one, you see there is a bifurcation here. These spots are known to be very rich in caveolae and caveolin. So it's a very interesting question to ask whether or how the presence of caveolin now affects the production of nitric oxide. And these are very recent data from our labs where we use caveolin 1 deficient mice. These mice have no caveolin 1, they have no caveolae. And you can see this is now the signal of nitric oxide produced in response to stretch by ventilation. And you can see there is an increase in the NO production here, which does not occur if the mice are deficient in caveolin 1. So this clearly is a very important structure in mechanotransduction. And I think we'll hear a lot more about caveolin in the next future. So now continuing with the plasma membrane. One other thing I would like to bring to your attention is that those molecules, the basic constituents of the plasma membrane, are not only bricks of the membrane, but they are very active molecules. And they serve the cells in a variety of ways. So I think this may be very well known to most of you. So for instance, from the phosphatidylcholine molecule, you can derive a number of very, very active biologically molecules, like deacylglycerol, if you cut here with a phospholipase C, inositol trisphosphate, if this is known inositol moiety, which is important in calcium signaling. This could activate protein kinase C, eicosanoids and plastic activating factor, if you have the phospholipase A2 in action. I think this has been known for a long time. What we are appreciating more and more is that also sphingomyelin, for instance, is a very important precursor of highly active molecules. And one of them is ceramide, and the other one is sphingosine. And both of them can be phosphorylated, and there is a great diversity in the actions of ceramide and ceramide 1-phosphate, or sphingosine and sphingosine 1-phosphate. But I don't want to go into details here, but there's a lot of focus on these molecules right now in the literature, and obviously these are very, very capable molecules. I will give you only one example for the sphingomyelin pathway. So what you see here is, again, that's a sphingomyelin molecule that you have been seeing. And there are two enzymes that can split this phosphocholine moiety, and there is an acid and a neutral sphingomyelinase, and now we just talk about the acid one, but that's not so important in this context. And you see then you get the ceramide molecule, which looks like this. And this actually is a very, very hydrophobic molecule. So this molecule is so hydrophobic, it can never leave the cell membrane. So if you would like to think about how ceramide is working, you have to consider that everything is taking place into the membrane. And now there is a recent increase in our knowledge of what this molecule can do. And ceramide has been linked to apoptosis. For instance, radiation-induced apoptosis, but also apoptosis of endothelial cells in the lungs in response to endotoxin. Then recently there has been a paper, I think in April or so this year in Nature Medicine, where they showed that a number of ways, but that ceramide is involved in the development of pulmonary emphysema. And we had a paper last year where we showed that ceramide could contribute to the formation of pulmonary edema. And based on that, we've also started to pursue that question further. One of the interesting things of this molecule is that the sphingomyelinases also be secreted from the cell, so you can measure it extracellularly. And together with a group of Professor Weiler and Kiel, we've started to measure sphingomyelinase activity in septic patients, and there clearly is an increase in those patients. And maybe even the sphingomyelinase can be a very attractive target because it's present extracellularly, so that's the extracellular enzyme activity. And if you remember, most of the sphingomyelinase is in the outer leaflet of the membrane, so it could very well be that this enzyme has to act outside the cell. And if an enzyme is outside the cell, it could be a very attractive target for therapy because you don't have to enter cells, you can get a much higher degree of specificity. So now I'm moving a little bit further from the basic constituents of the cells to the signaling that can be started. And I think this is a very nice example of how versatile the signaling can be. I think all of you will be familiar with G protein receptor signaling, and I come to back to this very briefly in a moment or later on. But this is a signaling mechanism. I think this is very innovative, so to say. At least it's very new for us. And also it has to do with physical forces that act on membranes. And there is good evidence to suggest, there are of course some receptors that can activate this pathway, but there's also good evidence that both pressure and stretch can activate this pathway. So what's going on here is, this is a metalloprotease. So this is a protease which is present on the outside of the cells. Okay, so it's inserted in the cell membrane, but the active site of the protease is outside the cells. And actually these proteases belong to a family that is called the Adam family, but I don't want to go into any further details here. Okay, so this is a metalloprotease. And this metalloprotease becomes activated by mechanisms that I don't want to go into now. That's not so important for us now, but it becomes activated here. And then cleaves off a substrate that is just sitting next to it on the cell membrane. And there are a number of instances, a number of things that could be here. One of them is the heparin-binding EGF epidermal growth factor. Okay, so then this protease cleaves off the HBEGF, and then the EGF binds on the EGF receptor again on the same cell. So I think this is really a very fascinating way of signaling. So most of them is extracellular, although there is some intracellular step, but the rest is going outside the cell and then going inside again. And it looks at this mechanism becomes activated when you over-distend cells or when you apply high pressure to cells. And I will just give you 2 examples of that, and just also show you, of course, some of the data you can get, and then talk a little bit how those data were gathered. So these are data from a paper in Nature last year from Chamberlain and the group from Jeff Drazen. And you see here in red the staining for the nuclei, and you see here in green, that's the staining for the EGF receptor. That's the receptor you've just seen on the last page. And they used 2 photon microscopy to look how the receptor is distributed also in 3 dimensions, and you can see it's present between the cells, and if you sort of wrap this up, you can see it's basically present in the lower region and not so much in the upper. So it's closer to the basal membrane. But that's so important here. So what they show them is if they use compressive stretch and have those cells in culture and use compressive stretch, then you see after 20 minutes there is an activation. There's a also maybe one step back. So this is, and I'll show you in a moment how this is done, this is a Western blot, an immunoblot using antibodies against the EGF receptor. And this is an antibody against the EGF receptor itself, and this is an antibody against the phosphorylated form of the EGF receptor, right? And that's a very important concept in actually all those signaling ways, that phosphorylation actually always ever plays a role. So here you can see this receptor becomes highly phosphorylated once it's activated. And actually what happens here is that binding of EGF leads to dimerization of receptors, so 2 receptors come together and then they become phosphorylated here, and then the signal transduction goes on. So one way to study whether these receptors become activated is to see, have they become phosphorylated? Okay? So in this way, measurement of phosphorylation here is a way to see, has this receptor become activated? And I know that many anesthesiologists are very scared when they see pictures like that. Because if you see talks in molecular biology, you see almost only pictures like that. And this can really be very annoying. But I think, of course, this is there are very important messages here. And most of you will be familiar how those pictures are made. But for those of you who are not, I will just give you a brief overview how these immunoblots are done. Okay, so this is also called Western blotting. Actually, it's called Western blotting because there was a guy called who was named Southern, and he started a way of hybridization DNA, and then later on we learned to hybridize RNA. That was called Northern blotting, just to make out a joke out of the original name Southern, and then we have Western blotting, just to continue this line of joking. Okay, however, of course, in papers also you find not often Western blotting, but now people more often write immunoblotting, but it's just the same. So what you do is you take your samples. So in this example before, you would take the samples from the cells at the different time points and work up the protein, and then put the protein here on the spot of this gel, and then you separate the gels, as you separate the proteins according to the to electrical charges along the gel. And then you take this gel and have another electrical device, and then you transfer the proteins that have been moving around here. You transfer the proteins from the gel to a membrane. And then next step, you continue with this membrane. Okay? And then you take this membrane, and the first thing is you have to block it with several albumin just to get rid of all the unspecific binding. And then you can start with a specific antibody against the EGF receptor or against the phosphorylated EGF receptor. Okay? And once this has been done, you use a second antibody that characterizes those domains of the IgG antibody here, and they have a light on them. And this light can be in a variety of ways. In this example, the light is labeled with an enzyme that produces a color reaction, and then you get this sort of signals here. And another example of a Western blot, which also is in this area, and which also relates to signaling by the EGF receptor, is work from the group of Snyder in 2002. And I think this is a very interesting piece of work. They used pulmonary epithelial cells, a cell line, and stretched them in culture, and then looked what happened until, in terms of activation of this very important signaling molecule. And I give you more information about this in a moment. So just at this time, ERK is a MAP kinase, and I come to this in a moment, but this is a very important signaling molecule inside the cells. So what they observe is that if they stretch the cells, as you can see, here. There is an increase in the phosphorylation of ERK. So again, like the EGF-receptor, also ERK is activated by phosphorylation. So if we want to see whether it's activated, we look, is it phosphorylated or is it not? So you can see stretch activates it. And interesting, what you can see here is that now if you combine stretch with this drug, which is an inhibitor of tyrosine kinases, you can completely prevent the activation of ERK. Okay? So this is another example of the pathway that I showed you before, that pressure or stretch activate this pathway. So there is release of endogenous ligands from the receptor, and then the signaling starts on, and then here you get to things that are also frightening to many people that are not very familiar with molecular biology. These are all these 3 letters abbreviations, right? So we have RAS, RAF, MEC, ERK, MAP, and so forth, right? And of course, and actually, if you want to learn this, it's good enough to learn the 3 letters, because the abbreviations are more or less meaningless. ERK stands for extracellularly regulated kinase. But I mean, that doesn't tell you more than ERK itself. So just don't try to understand what those 3 letter codes mean. Just take the 3 letter codes as a name of this molecule we are interested in. And so, most of these molecules that I will discuss now are involved in transmitting signals to this nucleus and to start transcription. So that is, if you think back at the boxes I had, now we have this box 2 and 3. And we have a very good idea now of how the DNA is constructed. So we have here which is called a transcription initiation complex, which is very similar for almost every gene. So we have the very famous Tata box, for instance, which is found just before the gene can be transcribed in all of the genes. So that's where the RNA polymerase is binding to this area. And then we have a core promoter region. And then, so that's where promoters bind, and I come back to the promoters in also these regions in a moment. And then, but even here, even up to 5, 6, 7,000 base pairs upstream of the genes, there can still be regulatory elements that have if molecules are binding to them, regulatory molecules, if transcription factors are binding to them, they still can affect the transcription. So it's very, very difficult really to get a good understanding how a gene is regulated, because there are so many transcription factors upstream of all the genes, of many genes. Okay? And the factors that bind to these particular regions, that recognize specific regions in the genes, and then change or activate those genes, they are called transcription factors. And the concept of that is shown in brief here. So we have a number of molecules. And this is a very typical cytokine receptor, like the EGF receptor that dimerizes upon activation. Then there is tyrosine kinase activity. And then there are a couple of steps in between. And this is really a very complicated example, so I left out a number of steps in between here. But the final thing is very much the same as for actually all other signaling molecules. So here we have RAF, which is a kinase. And then we have ERK and JNK, which are other kinases. And the whole idea of this thing is to activate a transcription factor that finally binds on the DNA. And one example here is AP1, which would be formed out of Jun and Phos, as a Jun and Phos dimer. But that's so important. The important thing to understand here is that basically it's a very simple pathway that goes from activation of the receptor, lots of phosphorylations of molecules in between, and in the end we have a transcription factor that then starts transcription of genes. And of course, there are also other names. So this, as you will see later, I think not see on the next. I'll give you one example first. And of course, this is again just to show you how this is relevant. For instance, to ventilating patients. Right? These are studies from rats in vivo that have been ventilated under different conditions. And you can see as an example of P-JNK, so phospho-JNK again, it's activated if it's phosphorylated. JNK stands for Jun kinase in this case. And you can see there is, as opposed to controls, there's a strong increase in the activation of this molecule. And I think you cannot see this very well, but we figured out that most of the activation occurs in alveolar macrophages, occurs in type 2 cells in the lungs. And another group, Deborah Quinn's group, showed that this actually is important for ventilator-induced lung injury, because they looked now at a molecule that is transcribed here if we activate this pathway and AP1. And one of the molecules is very interesting, is MIP2. And MIP2 corresponds, this is a mouse and rat analog to the human IL-8, interleukin-8, which is probably one of the most important chemokines, so to attract neutrophils into the lungs. So what they see is that if they have an inhibitor, first of all, if they have the same observations, they activate MIP2 gene. And if they have an inhibitor for J and K, there is no activation of MIP2. And there are further studies that show that this model this can be related to lung injury. So this clearly plays a role also in more complex models of ventilator-induced lung injury. So now one word to the MAP kinase cascade, because this is really involved in so many cellular responses. There's almost no cellular response without the MAP kinase system being involved. And of course, this creates a great deal of confusion because there are so many things where this is involved. But again, the basic concept is very simple. So we have three different pathways here. We have three final MAP kinases, the ERKs, the JNKs, and the P38. Right? And they become activated by different cascades of phosphorylation always, but the final step in the end is they all lead to activation of a variety of transcription factors that then turn on gene transcription. And unfortunately, since there are now a variety of transcription factors, and since many, many genes have binding regions for those transcription factors, it's not very easy to predict which genes will become on in reality. Even if you know that ERK becomes activated or J and K, that you still don't know really which genes become turned on because there are so many regulatory mechanisms. But I mean, that's the complexity we have to face. But I think this picture is very straightforward. So the whole idea is to transmit a signal to the nucleus to activate transcription factors that then starts transcription of cells. And actually, this picture is very similar for a whole variety of receptors, basically for most of the receptors we know. There are very little exceptions. One exception is the steroid receptors that resides in the cytosol. But even the steroid receptors then finally, of course, move to the nucleus. So what you see here, this is a gene, and these are all important transcription factors of a variety of different biochemically really very distinct pathways. So this is a gene, the G protein coupled receptors, right? And good examples for activators of those receptors is epinephrine, for instance, or TSH and so on. And of course, we know that mostly that they activate very, 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 very rapid reactions like glycogenolysis and so forth. But also activation of this receptor has long-term effects, and these long-term effects are also mediated or can be mediated by cyclic AMP, which is going to the nucleus, and in the nucleus there is a cAMP response element binding protein. And this protein binds cyclic AMP, and once it has bound cyclic AMP, this acts as a transcription factor, sits on the DNA, and starts transcription. A completely different class of receptor signaling pathway, but still with the same end, are the TGF receptors. So here it's activated by TGF itself, activins, and also BMP, bone morphogenic proteins. And this is a very simple pathway. So basically the transcription factor is very rapidly activated. There's not much steps in between. The receptor directly phosphorylates the SMAD protein, moves into the nucleus, and acts as a transcription factor. Then we have the pathway we already discussed before. This is a cytokine receptor pathway, which involves the MAP kinases. And here is another receptor, which is also very important in cytokine signaling. This is the IL-2 to 7, the interferon, GM-CSF. They all bind, or they all activate the so-called JAK-STAT pathway. And this pathway is much quicker than this one because there are much less steps involved. But in the end, phosphorylation here, STAT moves to the nucleus, again, activation of transcription. And then we have this example, which again has a similar step. NF-kappa B moves into the nucleus and then activates a number of genes. And NF-kappa B is really one, I could probably say, is the master transcription factor of inflammation. So almost every pro-inflammatory gene has an NF-kappa B binding sequence, and many, many of them are indeed activated through this pathway, at least partly through this pathway. And here, even though the end is the same, the story initially is a little bit different, because here there are also, of course, phosphorylation steps involved, but here the crucial thing to remember is that NF-kappa B resides in the cytosol normally as an inactive complex. It's bound to an inhibitor. So this is I kappa B, that's inhibitor of NF kappa B. And then the signaling works in such a way that activation of this receptor removes this inhibitor, and then NF kappa B is free to travel to the nucleus. And I think I have a very nice picture to illustrate that to you, that there really is a movement. And these are pictures from alveolar macrophages from mice that we have ventilated under different conditions. So this is an alveolar macrophage from a mouse under control conditions, and that's an alveolar macrophage from a mouse during overventilation. And what you see in blue here, that's the staining for the nucleus. And what you see in brown, that's the staining for NF kappa B. And you can see under control conditions, NF kappa B is in the cytosol. However, when these cells become activated, you can see now that the brown color moves to the nucleus. You have an overlap of brown and blue, and so you clearly can see really how NF kappa B moves to the nucleus. Of course, it's not always possible to get such nice pictures. So there has been other ways developed to measure translocation of transcription factors, which are really at the heart of signaling, I think, as we've understood now, to measure how transcription factors move into the nucleus. And this is a so-called electromobility shift assay. And since you also will see a lot of EMSA pictures, I will briefly explain to you how this works. So what you do is you first start by preparing a nuclear extract. Okay? So if you look back, you isolate the nuclei and prepare extracts of those nuclei. And that's what you work with. And of course, what you want to know is, has NF kappa B moved into the nucleus or not? So that's what you work with. You isolate the nuclei, make a nuclear extract. Okay. Make a nuclear extract and then do the following. So this nuclear extract now, that contains the nuclear proteins that you're interested in, and you want to know whether one of them is NF kappa B in greater abundance, are incubated with oligonucleotides that have normally a radioactive marker that is shown here, and then that have a different specificity. One specificity is indicated by the star, and this would be specific for binding to NF kappa B. So what happens now is that if NF kappa B is present in this nuclear extract, it will bind to this sequence that we provide externally. And once this has bound, it's much more heavier, it's much greater than the oligonucleotide without. So this means if you now have a GEL, this complex will move much slower than the unbound nucleotide. So it will be retarded here. So you have a shift in mobility of the DNA. And that's why it's called the electromobility shift assay. And the practical example is shown here. So this is now exactly what you see here. So if you see this down here, this is perfectly normal, right? So this is just the nucleotide, the labeled nucleotide that has been going through all the way. Okay? And here you see the sequences that have bound the NF kappa B peptide, so they are retarded. And here you see different conditions. You see that under control there is some activation. You see that both endotoxin and overventilation can stimulate that NF kappa B. And if you treat the lungs with steroids, you can prevent the activation of that. But I don't want to go into detail. This is just to give you an example how those pictures are being produced and how to read them. And of course, this is of great interest, I think, to the intensivists, because NF kappa B really is a master molecule of inflammation. And even back in 1994, there was a study from Ed Abraham's group that showed that NF kappa B is activated in macrophages from patients with ARDS. And we recently did a study where we ventilated healthy patients under different conditions. And the group that had a higher degree of stretching applied to the lungs, there also was an increase in the NF kappa B contents within the macrophages. And finally, and these are the last 3 or 4 parts now of this presentation, we have to talk a little bit about the histones. Because normally the DNA is wound around the histones and then packed very, very densely, as you all may know. So I don't know how many meters a normal DNA would be, but it's a couple of meters long. So we have really to wrap together this whole long DNA into the very, very tiny nuclei. And this is achieved by winding the DNA around those histones. And again, those histones are not simple bricks. They are not just by providing a structure, but they are very important for the regulation of transcription. And you can see here there are a number of different histones named H3, H4, H2B, H2A, and so forth. They sit inside here, and these proteins, they can be manipulated by phosphorylation, by acetylation, and by methylation. And there is now what we are starting to understanding a so-called histone code. And this histone code is very important because, maybe let's one go back, if the DNA is in this way wound around, it's completely impossible for transcription to start. Right? So we need this structure to pack the DNA, but if we want to transcribe, we have to get rid of the structure to dissolve and to loosen it. And the way to do that is, or one way to do that is, to modify the histones. And the most important modification here is acetylation of those histone moieties. Okay? And acetylation of those histone moieties usually turns a gene on. And this has to do with the charges. You eliminate some charges, and then the histone, this part of the DNA can unfold. Yeah? And here you see an example. This is a transcription factor. And if, for instance, NF-kappa B goes to the nucleus, it starts and it binds to its recognition site. This is not enough. This will not be enough for transcription to start. But what also happens is that these transcription factors call at the same site really a huge complex of a histone acetyltransferase, or if you want to stop signaling, want to stop transcription, a histone deacetylase. So there's a big complex, and this complex then, which is binding also to the transcription factor, is acetylating the nucleosomes, it's acetylating the histones, and then transcription is providing the prerequisite for transcription to start. There's also, just to briefly mention that, of course, other parts of this code, methylation usually turns genes off, and there is some discussion now maybe to what extent there can be epigenetic phenomena, so that whether you can inherit things you have acquired in life because the DNA has become methylated. Okay? And then there's phosphorylation also of the histones, but we're not really sure what this does at that time. And just finally, I would like to show you that also this is relevant for things we are discussing here. So we have recently started to look at histone acetylation in models of overstretching the lungs. And this is just a brief example looking at histone 4. And you can see that overventilating mouse lungs, stretching those mouse lungs, of course, leads to an increase in transcriptional activity, and by that also leads to an increased acetylation of this particular histone molecule. However, the story appears to be more complex, and this is the final data I like to show you. And even though it's a bit confusing, it shows that there may also be a great chance, if you understand these things, to achieve a very specific type of regulation. Because what we have done here is we used a compound which is called TSA. This is trichostatin acid, which is an inhibitor of the deacetylase. So if you prevent deacetylation, basically what you expect is you continue transcription for a longer time. So you would expect more cytokines, for instance, being produced. And of course, one prime cytokine that depends on NF-kappa B and it's turned on is tumor necrosis factor. And you can see there is, of course, an increase. This is now PCR. So this is the gene transcription. You can see there is, of course, a clear increase compared to control situation by overventilation. But now if we block the histone deacetylation, there is a very huge increase. So this is really what we would expect. And just as an aside, what has been shown is that steroids, one way how steroids act is that they recruit histone deacetylase to the nucleus. And this is one way how steroids can stop transcription. So if you block the histone deacetylase, there is, of course, an increased production of TNF, for instance, an increased transcription. However, surprisingly, this is only true for TNF. For interleukin-6, we have the completely opposite observation. So for interleukin-6, there is less transcription than for TNF, which shows that these two genes, which feature very prominent, and for most of the time we thought that these two genes are regulated very much the same, that they are governed by very different mechanisms in the end. And of course, this can also open a door for specificity if you try to intervene here. And just coming back to one question that was asked before, I think it could really be very important, also like Art said, to try to devise very specific ways to interfere with ventilation-induced side effects. Because we know when we start, and we know to some extent, we know more and more what we are doing when we ventilate patients. So I think it seems highly desirable to develop some very specific tools to intervene here. And I think molecular biology will help us a great way to do that. And finally, I hope that in the end now you know at least something about the constituent of the plasma membrane, of rafts and caveolae, of signaling by lipids, and by extracellular proteases, by transcription factors, and about histone acetylation. Thank you very much.